Dr. Pankaj, shall we continue? Shall we start? Pankaj, Dr. Pankaj,
Yes, sir. Yes, sir.
can I start?
Yes. Sir, yes, sir. So I invite Dr. J.S. Bedi, sir, Director, Center for One Health, to give the inaugural remark about the national webinar on Q fever.
A very good morning to all present in this national webinar. Firstly, I heartily welcome to our guest speaker, Dr. Deepak B Raoul, who is a renowned scientist in the area of zoonosis and food safety. And I also welcome all the participants in this webinar on Q fever, which is organized under the NEHEP IDP plan. Dear all, we know that Coxiella burnetii is a causative agent of Q fever, and it is considered as a ubiquitous zoonotic pathogen. It has wide variety of reservoir hosts, including mammals, birds, and arthropods, especially ticks. And the ruminants, they are considered as a major reservoir and important source for the transmission of disease from animals to the human being. And this disease is considered as an endemic in many countries, but the reports are limited. So it is considered as a largely neglected zoonosis. And this disease has been also marked as one of the most contagious and listed among list of global priority zoonosis disease. And it is also considered as a notifiable disease under the category of multiple species disease, infections and infestations by OIE. Undoubtedly, the disease poses a major negative impact on public health as well as on the socio-economic structure of the animal husbandry, particularly in the developing countries where the biosecurity measures and the diagnostics are need to be improved, especially for this rectial zoonotic disease. With these words, I invite Dr. Pankaj to introduce our guest speaker to the participants, and please start the session.
Okay, thank you so much, sir. So, yes, sir. So thank you so much, Bedi sir. Now it is my pleasure to introduce today's distinguished Okay, sir, am I audible, sir?
Yes.
Sir, am I audible?
Yes, Pankaj, you are audible.
Sir, sorry for the bad network here. So thank you so much, Bedi sir. Now it is my pleasure to introduce today's distinguished speaker, Dr. Deepak B Raoul. So Dr. Rawoul is currently working as senior scientist at ICR National Research Center on NEET, Hyderabad. Dr. Rawoul has more than 12 years of experience in research, teaching and extension activities, with the expertise on foodborne pathogens like Enterobacteriaceae family, Listeria monocytogenes, and Coxiella burnetii. Now, sir is well exploring the area of antimicrobial resistance with the expertise on antimicrobial peptides, phytocompounds as an alternate source of antibiotics. Dr. Rawoul has handled 6 projects as PI and co-PI from reputed international agencies and developed several techniques, including multiplex PCR for detection and lineage typing of Listeria monocytogenes, lateral agglutination test for enteroaggregative E. coli, as well as for Coxiella burnetii, synthetic LLO-based peptide ELISA for detection of Listeria. And sir has been awarded 2 postdoctoral fellowship one at Albany Medical College, USA, and the other at Institute of Medical Microbiology, Germany. Sir is also recipient of international fellowship under Young Biomedical Scientist category to undertake the AMR research. Dr. Raoul has published more than 60 international research publications in very reputed journal with high impact factor. And Sir has also guided 7 masters and 4 PhD students. And it is important to mention, Sir, it's always remain a privilege and great learning experience for me also to have completed my master's degree under your dynamic guidance. So with this brief introduction, I would like to invite Dr. Deepak sir to take over the stage. Thank you so much, sir, for sparing your valuable time.
Thank you. Thank you very much for that brief introduction. First of all, I would like to thank Dr. Bedi and Dr. Pankaj for inviting me for this lecture series at GADVASU. So I'm very ready to discuss some brief. But before I start with my lecture, let me tell you that actually Q fever in India initially has initiated by my mentor, Dr. S.V.S. Malik. So he is the key person, basically, who has started work on Coxella burnetii in India. And it is through his inspiration and guidance we have been just working on it, and probably will continue to work. Almost for 14 years we have initiated this work on Coxella. So I'll take you through my presentations, how we have initiated, but not much in detail because I know there is a very shortage of time. So 45 minutes what Pankaj has told me to talk about Coxella. So let me share my presentation initially, and also before I start, I see this talk is mostly for the students. I welcome all my students. I see some students, they are now right now in Gadwa. Is the slide visible to all? Hello?
Yes, sir.
Yes,
Yes, sir.
yes, Doctor.
Yes, sir.
Okay, so the today's topic is just to give you an overview of Q fever, and basically we all know this is a very important disease across the globe. But I try to restrict my talk from Indian perspective. So just to be brief, the right birth.
डॉक्टर पंकज कनेक्शन एरर आ गई कोई लग
हाँ यस सर मैं देखता हूँ हाँ सर का गया सर मैं एक बार सर से
कॉल
कम्युनिकेट
करिए
Yes, sir. There is no problem. Is connecting again, sir.
कनेक्ट कर रहे हैं हम
So sorry for the inconvenience. I request participant to stay tuned. Shortly, Dr. Deepak will join again.
Sorry for the inconvenience. That's
No issues, sir. Welcome back, sir.
नो प्रॉब्लम
No issues, sir.
we have that Wi-Fi connection here. So I'll just start it. So yeah, now is it visible?
यस यस सर
Yes, sir. It's going to say us.
गुड मॉर्निंग
Okay,
सर
so-
डॉक्टर बारबुदे सर गुड मॉर्निंग
गुड मॉर्निंग डॉक्टर जसबीर कैसे हैं?
Good morning.
बढ़िया सर बढ़िया सर
Good morning, sir.
गुड मॉर्निंग
Good morning, sir.
So I think shall I start, because already we have lost 5 minutes.
यस प्लीज
श्योर सर
Yeah. So
Sure, sir. No issues, sir.
so I was telling that this disease is known by several synonyms, but if you look back into the history, the disease was first identified somewhere in 1935 among the abatter workers. They were working in a large abatter at Brisbane, Australia. And from there, the disease was then later on people like Burnett and there were several people who have discovered this disease. I'll not name now much because I want to talk about the disease, but these were the people who later on identified. Earlier, this disease was not at all known to people. So that's why they named it as query fever, and that's why that name Q fever is still there, or abatter fever is still there as a synonym for this disease. So talking about the disease, this is basically an OI notifiable disease across different countries. And it is very highly infectious and contagious disease. And the reason why I'm saying very highly infectious and contagious disease, because if you see all the bioterrorism weapon, this is also considered as a bioterrorism ecological agent, Coxella burnetii. The infective dose required to infect a human is just 1 CFU. So one colony can infect a person, and that's why it is so highly infectious and contagious, basically because it is the most probable route of transmission is through airborne transmission. Of course, if you look at the mortality is not that much seen more in acute cases, but yeah, it leads to heavy mortality in chronic cases, particularly due to endocarditis. I'll talk about this disease when we talk about clinical signs. And if you see the recent 2012 ILRI report, that is International Livestock Research Institute report, this disease has been ranked among the 13 top priority zoonotic disease for our country. So this is a very important disease, but from Indian context, if you see this disease is said to be a neglected zoonotic disease. What could be the reason for this is basically, as Dr. Bedi during his initial talk, he also mentioned not much people are working on this, and there are some constraints with regard to its detection and diagnosis. So we will talk about that. So the disease is present across the globe, but except there are few countries where this disease doesn't exist at all, like New Zealand, Iceland, now French, and Polynesia and Norway. These are the few countries where disease has not been reported so far. So why this disease is so important? Because if you see way back in 2007 to 2012, that 5-year outbreak of Q fever, this was in Netherlands. It led to very huge economic losses. Nearly, if you look at, this was a paper from Nature, this was a news in Nature publication, and it says a tough lesson for Dutch Q fever outbreak. In this outbreak, nearly, I think 4,000, more than 4,000 humans were infected with this disease, and nearly 58,000 goats were culled just to stop the spread of this disease. And there are 3 different publications which have come up with calculation of economic losses, and every paper has different count. So I'll not go into it what exactly was the economic, but it leads to very huge economic losses. Talking about this pathogen as Coxella burnetii, basically it's a very small gram-negative bacteria. Now it's known as bacteria. Earlier it was known as rickettsia, and generally it requires this type 4 secretion system to enter into the host cell cytoplasm, wherein it later on goes to form, means it fuses with lysosome to form phagolysosome, and within the phagolysosome, it starts its replication. So once the infected cell is undergoes lysis, at that time, this bacteria is released from that infected cell, and then it goes to infect new cells. So generally, if you see into the generation time, it's not so that quick as E. coli, but it is around 20 to 45 hours for this particular pathogen. Yeah, this pathogen earlier was classified under family Rickettsia, because initially I said it was known as rickettsial organism. But due to certain characteristics of this particular pathogen, now if you see, this is the recent classification of this pathogen. It is classified under Legionellus, and genera Legionella, it comes under Francisella and Rickettsiella. Family is Coxellosis and genus Coxella, and this is all based on the 16S RNA sequence analysis. So now this bacteria, this is no more rickettsial organism, it's a bacteria. So this is very important for students to keep this in mind. And this is a typical teaching slide which always help to understand my students what is the difference between rickettsia and Coxella burnetii. So if you see the rickettsial organism, they grow freely in cytoplasm, whereas Coxella burnetii, they grow inside the phagosome, which is present in the cytoplasm. So that is the difference between rickettsia and Coxella burnetii. If you see the phase variations, you don't see in other rickettsial organisms, but this is found very common in Coxella burnetii. There are 2 phase variations. Phase 1 that is very seen in host. Of course, phase 2 is also seen in host. But if you isolate this pathogen from a host during that phase 1 stage, and if you keep on passaging in the lab, then it eventually goes into phase 2. So normally, if you look into that criteria, phase 1 is said to be a virulent phase of Coxella burner type. Phase 2 is avirulent. I'll discuss this later on. Transmission is basically for rickettsial organism, it is vector borne. But for Coxella, there are several modes of transmission. The next slide will show that. So it can be transmitted by more routes, but even ticks are involved, and even the major route of transmission for Coxella is aerosol. Whereas in case of rickettsia, the major mode of transmission is basically vector borne. So these are the different diseases under rickettsia, like spotted fever, Rocky Mountain spotted fever, typhus. In typhus, we have epidemic, endemic, scrub typhus. And in Coxella, we all know Wells-Felix reaction is negative for Coxella burnet, and that was the reason why it has been again classified under bacteria, whereas all rickettsial organisms, majority of them show positive reaction in Wells-Felix reaction. That is wherein they use this protease antigen for that agglutination reaction. So this is a basically potential pathway for Coxella burnet transmission. So if you look into the life cycle of this pathogen, this is basically maintained in the sylvatic cycle. But now we have another two more cycles. That is, one is the domestic cycle, and there is one more, which is not very commonly seen, but this is a predatory cycle. So the pathogen is maintained in the sylvatic cycle only when the domestic animal come in contact with this sylvatic population like wild birds and mammals. When they come in contact, maybe either through airborne transmission or wind dispersal, the cattle or the ruminants, they are infected. And as Dr. Vedi rightly said, these are the major reservoirs for this particular pathogen. And from domestic animals, then it can pass on to the environment. And from environment, there are three different modes of transmission, either by direct or indirect contact. That means if you come in contact with contaminated material from an infected animal like placenta or aborted material, if you come in contact with hides or wounds, or the contaminated urine, manure, or even body secretions, that pathogen can easily pass on to humans, or through ingestion. As you know, this bacteria is excreted for a very long period in milk. So if you consume those milk which is raw, probably that can lead to infection. Then even inhalation, this is the most major route of transmission for basically occupational workers, like inhalation of aerosols. Because basically if an animal is aborted and if their placenta is not properly disposed, or their secretions or their manure, it is just thrown in an open environment, then those infected aerosols can infect you through this airborne transmission. And besides this, there are other modes of transmissions like blood transfusion, even through infected parturient women, or sexual transmission. Bioterrorism mode is there. So these are other probable mode of transmission to the humans. So these are some of the modes of transmission of this disease. And what are the risk factors that are associated with this disease, particularly in humans? If you look into it, there are two important risk factors. One is animal-related factors, and the other one is environment and behavioral factor. The animal-related factors, basically, these are the people who are in constant touch with the animals, like the farmers who are in direct contact with animals, either for their milking or when they are assisting calving. In this case, even the veterinarians can be at risk. Or if you are handling a placenta without taking proper precautionary measures, or the people who are shoveling manure or feeding the animals, or even now, if you see recent literature with this regard, with this pathogen, people who are petting animals, they are also at risk, because even there are reports that pets have transmitted this disease to human population. And with regard to environmental and behavioral factors, basically, when we talk about the environment, it is the environment where the animal is housed. If the house is not proper ventilated, then the disease can spread very rapidly to other which are not infected, they can be also infected. So if it is poor ventilated barns, then that disease can spread very rapidly, particularly this Coxella. Then another is like occupational hazard. This disease is classified because you see the disease is very commonly reported in farmers, vets, even in abatter workers. We are working on that particular aspect here at NRC meet. And then even other environmental or behavior is the consumption of food or the cheese that or the raw milk. If it is not pasteurized, then that food can be a potential source of infection to the humans. Of course, then other behavioral factors like having addiction towards smoking or tobacco, drinking alcohol, this can also predispose the individual to this disease. So these are some of the risk factors that are associated, and we have investigated it. I know Dr. Pankaj has worked on this, and he has worked very, I think, meticulously, particularly to find out the actual risk factors for this particular disease during his PhD work. So what happens in humans when once you get this pathogen, probably say through aerosol routes? There are two clinical forms that you see for this particular disease. One is the acute Q fever and the other one is the chronic Q fever. But if you see, this chronic Q fever are very less. The acute Q fever accounts for most of the cases, and out of this cases, nearly 60% of the cases are asymptomatic, while 40% of the cases may show symptoms, but out of this 40, again, 98% the symptoms are very mild in nature. So this disease often go undiagnosed in human population. Only 2 to 5% of these acute Q fever cases may show some severe symptoms like fever, pneumonia, hepatitis, then some chills, fatigue is there. So these kind of symptoms are seen in those infected individuals during acute Q fever, and sometimes they may need hospitalization. But for example, if this 2 to 5% of severe patients, if they have some predisposing factors like immunosuppression or pregnancy, heart valve lesions, or vascular abnormalities, then such patients, acute fever can transient itself to chronic Q fever. And in case of chronic Q fever, the fatality rate is very, very high. It's around nearly 65%. And the major cause of the death is endocarditis. Of course, besides this, there are other clinical implications like chronic fatigue syndrome, osteomyelitis. Even you see repeated abortions in case of pregnant ladies, or sometimes chronic hepatitis. This kind of disease are also seen with regard to Coxella burnetii infections in humans. In animals, of course, the disease is prevalent, and these are some of the information with regard to seroprevalence from different domestic animals. I would like to give you information that in cattle it can range up to 82%, sheep and goat around 73%, dogs around 22%, and cats it is say 19 to 42% prevalence that has been reported. This is not from India, this is global seroprevalence reports. But in animals also, initially most of the infections are of subclinical nature or asymptomatic, but later on you find in these animals, if they are pregnant, it may lead to abortions. And abortion rate is around 5 to 91%, 5 to 91% in case of infected small ruminant flocks. As far as the prevalence of Coxella in cattle is concerned, if you see the herd level prevalence is very, very high. It's around nearly 32 to 94%. And these cattle are the major risk for any humans as well as for animals, because they keep on excreting this pathogen in the milk for a very long period. I'll just show you the information. This is a paper wherein they have seen the kinetics of this shedding of Coxella burnetii. And if you see a cattle, it can excrete this pathogen for nearly 13 months. which is a very long period, almost more than a year. Whereas the other species like goat and eve, they don't excrete this pathogen to that extent. Of course, in case of sheep, you find that this pathogen is excreted in vaginal mucosa for 71 days. So basically, urine basically of these infected animals are the potential source of infections to humans. This is a slide which gives you an information, particularly about the prevalence of this pathogen across the globe, and you can see nearly 5 continents have reported this disease, and it is said to be endemic in almost more than 51 countries across the globe. Even India is said to be endemic for this particular disease. And I'll just give you some brief studies what India has carried so far. Most of the studies are from our lab, but there are few studies which have been carried from other parts of the country, particularly in humans from CMC Vellore. We have people working there. Even GADVASU is working on this Q fever. And I know that the person who are working on Q fever from Tanua's, Dr. Raja, he's working, he has an ICMR project on Q fever. And of course, our lab at and all students, they have initiated their work on Coxella, and this is the data generated through their studies, or either through their master's program or PhD program. And we have reports of this Coxella from humans, animals, as well as from wild animals, but this earlier studies were mostly based on seroprevalence. So the studies never thought of going for detection of this pathogen using molecular tools, because we all know there were no molecular tools during that period. So most of the studies were zero-based studies. But now if you see the recent studies, maybe say after 2010 onwards, if you see the reports that are pouring out from India, they are basically either based on molecular studies or in using some new serological tests like ELISA, which is recommended even by European Union. So these are the studies are there, and these are some of the studies which I was telling. Most of the work is they have different animal populations like cattle, buffalo, sheep, goats, and prevalence has varied based on the student that has taken that work. Several students have worked on this, and they have employed all possible techniques to detect this pathogen, like RT-PCR, real-time trans-PCR, common PCR. These are the specific PCR that are used for Coxella burnetii. IFA, we all know this is the gold standard test for humans, and ELISA is now recommended over CFT. CFT is the gold standard for animal population, but now ELISA is recommended to screen Coxella, particularly serology. Of course, isolation, no doubt it is a gold standard, but it's very tedious. I'll come to it later on. And we have also developed some lab-based tests, and we are in that process to optimize so that we can soon have a very convenient test for field use. So we have even worked on humans, and in humans, you can see the prevalence has been reported, particularly with spontaneous abortion in humans. We have prevalence around 25%, and we could isolate 4 pathogens from that. And even we have reported some positivity rate in abatter workers, vets, farmers. These are all reports from IVRI, but even at NRC meet, we have done some seroprevalence studies in abatter workers, and there also the prevalence rate is more than 21% in abatter workers. So that's a huge with regard to this disease. And even we have screened some foods at IVR, and there also we have found this pathogen. Of course, for students, everybody knows now this is an index organism for pasteurization. So this pathogen is very, very important, and it's very sturdy as compared to Mycobacterium, and that's why it is an index organism. So I think this, I have kept this slide not just to show that this is the publication, but this is the recent publication from India that has come out. This is from our group. with the initiative of Barbade sir, we have published this review in Environmental Microbiology Reports, and it talks about the entire occupant studies that are carried so far from India. And it's not only from India, but the other countries surrounding India, that is the Indian subcontinent. So it is a very comprehensive mini review for Coxella, and those who are interested, they can just go and download this. So it will give you complete information about the disease. And with regard to India, we have reported from the papers have been collected right from the first case that were reported from India till 2021. Sorry, 20. We have documented the occurrence of this Coxella in our country, particularly from sheep, goat, humans, camels, cattle, buffalo, every possible vector that is involved in transmission of this disease. These are some of the other studies from our group, particularly I said Dr. Dhaka has worked on risk factors for Cellulosis during his PhD power. There are other students who have worked on different aspects of Coxella in animals. We've also seen the role of ticks in infected cattle for Coxella. Of course, in our study, we couldn't find any tick positive, but we can't say that ticks are not involved in transmission of this disease. We have to investigate more thoroughly, and that in a very systematic manner. And now coming to the detection, which is why this disease is so neglected, because when I'll discuss about the detection of Coxella burnetii, you yourself will understand why it is so difficult to go and start working with Coxella, because there are actually different methods for detection. First is the isolation. So you can do isolation either by in vitro methods or in vivo. There are some models. In vitro, these are the different cell lines that are used, like Varroa cell lines or buffalo green monkey cells, or human embryonic lung fibroblast can be used, wherein you can try to isolate the pathogen. In case of in vivo, we have guinea pigs are well known. These are the first lab animals. And if you infect your material into guinea pigs, probably you can see that if the guinea pig shows some increase in temperature after 5 to 12 days post inoculation, you can suspect that there may be Coxella, and you can just take out the spleen of that guinea pig and go and culture either in cell culture based media, or if you don't have guinea pigs, you can go for mice, but all mice are not equally susceptible to this Coxella burnetii. It's better to go with some AJ mice. These are highly susceptible to infection, wherein you can see these mice can show mortality up to 100, morbidity up to 100% and 70% mortality. Of course, if you have some ethical issues with regard to laboratory animals, then you can think of even going with embryonated egg. It's a 10-day-old embryonated egg, what is required, and how we have to process your sample. You have to first initially prepare your inoculum using a differential centrifugation method. This is what it is given here. And then you have to prepare your inoculum and then go for chick embryo inoculation by yolk sac method. And after certain incubation, maybe after 5 to 10 days, you start getting some lesions on yolk sac membrane, and even the embryo dies because of this infection. And what you see, if it is an infected yolk sac, this is from our lab pictures, you can see that the yolk sac membrane can show tinge of hemorrhages, whereas this is towards your left hand side, it is a negative control, but this is the infected. You can see there are red spots. And once you find such lesions on the yolk sac membrane, then go and prepare a smear. And after preparing a smear, you can't use gram staining for this particular pathogen. The best staining is the Gemini staining, which is recommended for Coxella burnetii. So go for Gemini staining, and you will see pinpoint pink color dots like bacteria with a green background. That is what you often see. Here in this figure, you don't see green background, but yeah, there are some pinpoint pink color dots, which are indicative of Coxella burnetii. So this is how we generally go for isolation, but with regard to a diagnosis in animal is concerned, what are the lab tests that are recommended? Of course, we all know why CFT, but when we talk about CFT, the test itself is very cumbersome to perform, and it lacks that good sensitivity as well as specificity. And that's why most of the people don't think of using this test for daily routine diagnosis of this disease. So what is recommended by European Union, and even if you look into the literature, most of the people recommend for ELISA, and it is said to be far more sensitive and specific in comparison to CFT. Of course, if you want to look for acute cases or shedders, because we are looking for the antigen, and antigen you see in case of this disease is seen during the early phase of the infection. So during the early phase of the infection, if you want to identify the shedders, then generally PCR is the best tool. And there are different PCR. The most widely used PCR is the trans PCR, wherein they target this insertion element, trip 1111. And if you look into the genome of this particular bacteria, there are almost 7 to 110 copies of this insertion element. And it happens to be the important target for Q fever. Of course, besides this, there is an outer membrane protein wherein people go and target, that is the COM1, that is the outer membrane protein of this pathogen. It's a single copy gene. So in real time, generally people target this COM1 PCR. So there are different versions of PCR for detection of this pathogen, like COM1 PCR, they are real-time PCR. Also, even in real time, we have Tachman chemistry PCR from our labs that have been published. But what is now recommended to acute or true shedders is just go with a combination of PCR as well as ELISA. Because what happens, this antigen remains in the body for a very short period of time. So after the antigen, once you have that antibody coming into the host, this antigen is cleared from that infected individual. So what is left is antibody. So a combination of PCR and ELISA is the best tool to diagnose this Q fever for identification of this infection in animal folks. So in these connections, we have also come up with certain different tools, and these are the papers that we have published. We have developed a lab-based for detection of this Coxella, and these are very recent publications. The work is performed by Manish Kumar, who is now assistant professor in Luas, and JP Yadav. Both have worked on different antigens of Coxella to develop a LAT-based test. As you all know, LAT is a very handy test which can be done at field level. So we are working on that, and now we have come up with really good important results. I don't want to disclose those results, but that work was done by Dr. Neha. She is now pursuing a PhD at GADVASU. So she has done really wonderful work, and we have come up with a very high sensitivity and specificity for this LAT format using a very modified version, that is a modified antigenic peptide of Coxella. So once the patent is filed, the publication will soon be there. This is for the students, just to for a curiosity, what is the treatment in humans? In case of acute cases, doxycycline is the drug of choice, and you can go twice a day, 100 mg for 14 days. And in case of chronic form of this disease, you need a longer medication, almost 2 to 3 years of medication. But the prognosis is still guarded in case of disease. If you are infected with this pathogen, it is said that you will develop a long-lasting immunity against this pathogen. As far as the vaccine is concerned, this is the only vaccine that is existing in the globe, and it is there in only Australia country, and it is an inactivated Coxella burnetii vaccine for humans. prevention and control, basically what we can do, and these are just general hygienic and sanitary practices, wherein in animals, what we can do is, if you're importing animal, it's always better to quarantine, keep them separate, see health status, if there are animals which are pregnant, and if at all they go abort, don't just attend those aborted animals without any proper protection. Just try to have a proper PPE and then attend those animals. If there is any placenta or anything like their excretions, they have to be disposed of very properly. And of course, if you see any tick population in your farm, then you have to adopt certain measures to control that tick population. In case of humans, of course, you don't consume anything raw. Always, particularly in milk, as I said, the pathogen is excreted for almost more than a year. So it is always better to use or drink something which is pasteurized. And of course, for example, this disease is still not so popular among veterinarians. So it is a time for all veterinary public health people to educate these professionals or the farmers, or particularly to have some information about this disease. Of course, at IVRI, when we were there, we had developed certain leaflets for awareness among the population, because nobody knows about this disease to that extent. Even at NRC meet here, awareness programs on occupational zoonosis, particularly among abatter workers, because this is again associated with occupational disease. We create awareness, and we have one more project running wherein we are trying to identify emerging abatter associated zoonosis in abatter workers or in abatter value chain. And there we are not only targeting Q fever, but there are other which are not known, but these are reported in a huge amount, means the prevalence is in a huge amount, like chlamydia, hepatitis, the CCHF in abatter workers. So we are also targeting those diseases in abatter workers to see whether this infection really exists in our Indian conditions. So this is for all the students who really want to work on this particular pathogen. So there are lots of research options available. First of all, let me tell you that in India, we don't have that much systematic studies available. So a very systematic and large-scale multi-centric study covering different agroclimatic zones of our country are required, particularly in different herds or small ruminants, even pet animals, they are a potential source of this infection to humans. So those areas are still lacking from Indian context. Even the role and association of this Coxella burnetii in causing reproductive disorders or mastitis in bovines or in caprines is still not known. And what are the risk factors, that is still not known. So maybe that can be explored. Even we don't know what are the circulating genotypes in Indian population or in animal population. So that has to be taken care of. There is still, of course, we are working on this particular aspect, like we need an easy test to perform, particularly or preferably a pen site diagnostic test. And finally, I would say the awareness among vets and medicos needs to be created, because this is still a neglected disease. We have to educate what this disease, how it spreads, and what measures we can take to control this disease. And very important, I see Dr. GADVASU team is working on this particular aspect, economic impact of Q fever, and this has to be really studied, because it leads to huge economic losses when there is an outbreak of this disease. So these are some of the research options wherein students or faculties can think of working on these different aspects of Q fever. And before concluding, I would like to thank all my mentors, particularly Dr. Malik sir, Dr. Parbade sir, and all my students who have worked very meticulously for this Q fever. To name few, Dr. Pankaj, Dr. Manish, Dr. JP Yadav, Dr. Neha, Dr. Vinod, all they have been working meticulously again for this pathogen. So I would like to thank them for contributing their slides for this presentation. And finally, I would like to thank the organizers for giving me this opportunity to talk about Q fever. Thank you very much.
So thank you so much, sir, for your kind mention, and you have been always motivational for us. Sir, thank you for the comprehensive overview on the Q fever, its etiology, host range, transmission cycle into humans and animals, and putting crucial insights, especially in diagnostics. That's really very difficult and challenging. And most important about the control and prevention measures, especially on the awareness aspects, because most of the time even physicians and veterinarians are not well aware about the epidemiology of disease, especially in Indian context. So now I request participants to clear any doubt if they are having on this platform with our guest speaker. So you can put even your queries in chat box. So sir, I can see one query from Dr. Hina. That the media you mentioned for cultivation, is it in use in India anywhere?
Yes, let me tell you, this media has come up very recently. This is an axenic media for cultivation of this Coxella burner type. But what is requirement beside this media is a specialized incubator, wherein we need a 2.5% controlled release of oxygen and 5% CO2 and rest other nitrogen gas. So this incubator is very difficult to fabricate. Of course, this media has been this incubator was fabricated at Nagpur Veterinary College at Department of Veterinary Public Health, because that is a college which is working on this Q fever. But yeah, this media can be a very handy tool for isolating this pathogen.
Okay, thank you, sir.
It is used in other.
Thank you, sir.
Thank you, sir. So is there any doubts? Or one more question, what are the chances of availability of simple and rapid test for diagnosis like we have for brucellosis?
Well, this answer can be even given by Neha. There are very bright chances we will be having very soon diagnose brucellosis, we will be diagnosing Coxella also.
Okay, thank you, sir. Is there any doubt? You can unmute yourself and can directly ask to the our speaker if you are having any doubt. You can unmute yourself and you can ask directly, or you can put in chat box.
Hello.
Yeah.
Good morning, sir.
Good morning.
Sir, it's a very nice lecture. Thank you for sharing your views to us. Sir, I want to ask one question. Is there any vaccine available in India for the Q fever?
No. I said there is only one vaccine so far that is available for humans only in Australia. There is no vaccine for this particular pathogen against animals, neither for humans in India.
Thank you, sir.
Thank you, sir. So is there any doubt? You can directly unmute yourself and ask, or you can contact to sir anytime. We will provide you the mail of the sir.
And sir, it was very great to listen to you. And your lecture was very informative, interesting, and very well prepared. I must congratulate to you and your team members, sir.
Yeah, as I said, all team members are our students. So I think that congratulations goes to the students who have helped me in making this presentation. So their work has made this presentation. So I congratulate all my students for that.
Yeah, thank you so much, sir. And this is a very good, I think, platform for the PG students. Because sir has given a very detailed insight about the Q fever, and he also opens the future perspective of what we can do in the Q fever aspect. And I think the undergraduate and postgraduate students, they should think about on these points, and they can make their mind to study in future related to the Q fever. because I think the sir has a very good experience. Their lab is well equipped, and even GADVASU is also working on a Q fever. So since though it is a neglected zoonosis, but we can explore further lot of things on Q fever. Like I asked a question to sir, what are the chances of simple test? Because sir, in many times we are encountered with the human patients who are have a symptom similar to Q fever and or brucellosis, but they become negative to brucellosis.
Yeah,
Although they have
true.
occupational exposure, but we are not able to diagnose Q fever because of the lack of this rapid and simple test.
Yeah.
But I hope that your team will develop in a short span of time, and it will be very widely practicable use in the field conditions.
Yes, sir. Actually, this test has been taken forward even in One Health project here, which has been initiated by DBT. So we are a part of that, and this test will be validated across the country to ensure that this test is really working in field conditions. So once we have that, probably that will be very handy to diagnose the disease. Because this is again very neglected. The disease is there in the population, but still we are not able to identify those infected individuals because most of them are asymptomatic.
Sir, we are having now Sandeep sir also, sir. Sandeep Chaudhary sir. Welcome, sir.
And yeah, Sandeep sir is also working on that aspect. Students are also working a lot on Q fever.
Sir, Dr. Barbuddha is there?
I think he was there earlier. I think he has left because
I'm very much there.
Okay, sir. So, sir, we want to listen your remarks on this lecture, sir. Please, sir.
As usual, Dr. Deepak has given excellent presentation, and since I am observing him since his student days, when you are doing MBSc, since then we are associated, and now we are working on this, and we want to what brucellosis test by many people they work and very easy to diagnose. Like that, now we are working on Coxiella, of course, Listeria also we are coming, Chlamydia also we have started now, and also now 2 days we are one of our students working on Bartonella also. So we want to come those platforms, and we want to tie up with as many companies and commercialize as early as possible. But this actually particularly Coxiella, we got very good leads. So we want to have IPR that protection. Already we have prepared and given the proposal at IVRI for filing the patent. Once it is done, I think we can commercialize it and come with soon with the latex aggregation test. Even lateral flow assay we are working at NRC meet now. My colleague, they have started some work. We have some facility also now here. So we want to come for both the that formats, LFA as well as latex aggregation test. So I hope another maybe 6 months or maybe a year's time, we'll have a lot of diagnostics at least for Coxiella to come up. And these are very specific and also sensitive, so that problem can also. As far as the isolation is concerned, we are also facing problems, and that media actually is not available. But we found out the composition of the media, so it is available. But that incubator is again tricky. We need control. You need to control oxygen. You need to control CO2. You need to control nitrogen level also. But let us see now if you are some companies we are working, and they have already 2 companies. They have such type of incubators. So I think I want many and more and more people should work on this aspect. While working in Chhattisgarh, I think we have not shown the slide here. So that slide particularly, we have done metagenomics of ticks. So we found one of the sample, 58% of the microflora was only Coxiella. So much prevalence is there in Indian concept, but many people. Secondly, we got lot of positive cases in humans also. So particularly infectious endocarditis, in which are culture negative cases, maybe tried for Coxiella. Then even spontaneous abortions in humans. I think these are the 2 where everybody can start working on this. Thank you very much.
Thank you so much, sir. for sparing your valuable time for giving your remarks on this lecture. Now I invite Dr. Pankaj to pay a vote of thanks to the guest speaker and Dr. Barbudi sir.
Thank you so much, sir. So it is the time to conclude our today's national webinar. So I put my special thanks to Dr. Deepak B Raoul sir and Dr. Barbudi sir. Even Sandeep Chaudhary sir also have joined us to sparing their valuable time and to motivate us or our participants to do further research on this important pathogen. I want to thank our dynamic director, Dr. J.S. Bedi sir, and our dynamic dean, Dr. S.P.S. Guman sir, and even the IDP cell members to render all the facilities for smooth conducting of this national webinar. So, and more especially to the participants for their active participation and to stay tuned throughout the lecture. Thank you so much all. Thank you, sir.
Thank you, sir.
Thank you, sir. Namaskar, and
Thank you.
have a great day.
Thank you so much, sir.
Thank you so much, sir. Thank you, sir.
Okay,
thanks to all the participants.
Yes, sir. Thank you so much, sir. Thanks. So, sir, we can end the meeting, I hope, sir. Okay, sir.